Hello everyone. In the part 4 of RNA-seq analysis, we applied the upstream analysis workflow to predict master regulatory molecules that play a role in triple-negative breast cancer cells. In the part 4 of this video, the results of master regulator search was shown, and today I'll focus on the results of promoter analysis. The output includes a folder with a summary table, yes and no promoters, and yes and no optimized track files. To begin with, I open the summary table in the workspace. Each row summarizes the information on one positional weight matrix. For each selected matrix, the column yes density per 1000 base pair, no density per 1000 base pair show the number of matches normalized per 1000 base pair length for the sequences in the input yes set and input no set. The column yes-no ratio is the ratio of the first two columns. Only matrices with a yes-no ratio higher than 1 are included in the summary table. The higher the yes-no ratio, the higher is the enrichment of matrices for the respective matrix in the yes set. We can also see the matrix cutoff or model cutoff and a p-value for each matrix. These are the matrices or site models with the matches overrepresented in the promoters of genes upregulated in breast cancer triple-negative cells. Here I selected rows with fold change greater than 1.3 and I open the details of the genes upregulated in triple-negative breast cancer cells, and for each gene of our interest we can see the structure of the promoter. Here each row represents promoter on one upregulated gene. The column sites view shows the schematic representation for the matches of different matrices. The column total count shows the number of transcription factor binding sites for all matrices together. The next columns are named as matrices and the numbers show how many matches for each matrix are there in a particular promoter. If I am interested to know more details about each particular match, I can find that information in the info box below. We can see the details about the sequence, the position of the binding sites, the score, the core score, and the matrix logo. From the previous part of the analysis, I know that some of the transcription factors that regulate upregulated genes in triple-negative breast cancer cells are upregulated themselves in the same conditions. Here I am interested in transcription factor IRF1. It is highly upregulated in triple-negative breast cancer cells versus ER-positive cells with a log fold change of 3.0. I am interested to know IRF1 target genes, and I can sort this table by IRF1 matrix. These genes, TNFSR10B, EPSTL1, TAP1, PRKCQ are regulated by IRF1. The IRF1 target genes can be selected and saved as a subset for further study. A total of 54 IRF1 target genes are identified that are upregulated in breast cancer triple-negative cells. Next, I am interested to visualize the identified transcription factor binding sites on the genome. I open the track file Yes Promoters in the inbuilt genome browser. Yes Promoters means the promoters of the upregulated genes in my study. The track Yes Sites Optimized when opened in the genome browser visualizes those sites that are overrepresented in the promoters of the upregulated gene. As shown here, for the promoter ERRFL1, the length of the promoter is 1100 base pair. All the transcription factor binding sites can be seen binding to the promoter and can be visualized in detail here. They can be seen as a complete list from the Sites tab below the workspace. To summarize, in this part I have shown the results of the promoter analysis. 437 promoters of genes upregulated in triple-negative breast cancer cells were analyzed. Transcription factor binding site enrichment identified regulatory transcription factors, and I have shown the visualization of identified transcription factor binding sites on these promoters. Thank you for watching and do subscribe for more updates.